Welcome everyone to today's research announcement. We're really excited to announce this year's research grant competition results. On behalf of Muscular Dystrophy Canada, welcome and thank you, I should say right from the beginning, for your ongoing support. My name is Hamira Osman. I'm the VP of Research and Public Policy at MDC. As I just mentioned in the chat, I would like to again remind you that today's session is available in English and in French. There's an interpretation icon at the bottom of your screen. You can go ahead and select English or French, the language of your choice. We're really pleased to be here today because today's research announcement is really another testament to our strong commitment to research. We recently launched our strategic plan. We're actually in year one of our 5-year strategic plan. And you can really see that expanding research infrastructure, improving public access to healthcare, information, education, and other community resources is a strong commitment here at MDC. And when we even look at the last year alone, we had seen an increase, 48% in funds directed towards research initiatives. This really helps us to build on collaboration, outcomes, and research program continuity. When we think about our investments with research, we're thinking about our investments in research projects like the ones that we're going to learn about today. In people, the early career researchers, the trainees, the scientists, the clinicians who help to conduct this work. The partnerships that are built around these research projects or that are built as part of NMD4C, the Neuromuscular Disease Network for Canada, and other partnerships that really are about supporting clinical and translational research or supporting policies and using evidence to really influence positive change. With that, I wanted to go ahead and invite Dr. Robin Parks. Dr. Parks is the chair of our Medical and Scientific Advisory Committee. He's an associate professor and senior scientist at the Ottawa Hospital Research Institute. And Dr. Parks will really speak to the process that was involved in selecting the recipients of this grant competition. Over to you, Dr. Parks.
Thank you, Hamira. It's a pleasure to be involved in today's webinar. I just wanted to say I have no disclosures or competing financial interests. So Muscular Dystrophy Canada supports about 150 different neuromuscular disorders. And for many of the disorders, there's still many things that we don't know. For some, we don't know the causative gene. We don't understand the molecular mechanism of the disease. We don't have adequate animal models or cell culture models to be able to create or test new therapeutics. We don't necessarily understand the natural progression of the disease in patients. And we don't necessarily know the best practices for managing symptoms in patients with neuromuscular disorders. And there's many other things that we need to uncover. And this is where MDC comes in, because MDC is dedicated to supporting world-class research to solve some of these unknowns. And there's really two types of research that are supported through MDC. The first is the translational science seed grants. And so really those are lab-based studies to understand the molecular mechanism or develop new therapeutics. And that's testing in cell culture and animal models of these neuromuscular diseases. The second is the clinical research grants. So this is research to improve treatments, clinical care, quality of life, or standard of care. And of course, these are patient-based research. Progress really requires both translational and clinical research. And the example I'm going to give you is the development of nusinersen for spinal muscular atrophy. So SMA, of course, is a devastating neuromuscular disorder that fortunately has a development of several new therapies recently. And this is really the timeline for development of this. So work began in the clinic in identifying and at first describing the clinical nature of spinal muscular atrophy, and then transitioned into the lab where we identified the gene responsible for it. We developed mouse models and started testing some of the therapeutics, which then moved back into the clinic to be tested in people and eventually gave rise to the therapy that we have today. And this is really a simplified timeline of what's gone on, because really there's been thousands of researchers and thousands of individual projects that have each contributed to this. So each of these small projects provides a piece of the puzzle, the 1,000-piece puzzle that's necessary to eventually come up with an effective therapy for neuromuscular disorders. And so that's what MDC is addressing with these projects, is trying to provide a new piece to the puzzle so we can move things forward. So how does MDC decide what research to fund? Well, we rely on peer review. And so this involves grants are reviewed by an expert panel of 6 to 8 scientists and clinician researchers, as well as a lived experience reader. And really, we need to thank these people for taking the time and their expertise to contribute to the grant review process and make sure that the best grants are funded. So this panel critically evaluates each grant, the goals, the strengths, the weaknesses, and the potential impact. And the applications that are deemed highly meritorious and likely to have significant impact in the neuromuscular disease community forwarded funding. Also importantly, unsuccessful applicants are provided constructive feedback on how to improve the application for resubmission in the next competition. So hopefully it will be successful down the road. So what happens once the grant is awarded by MDC? Well, over 1 to 2 years, the investigators complete the goals of the grant, moving the science forward and advancing our knowledge and to drive progress and innovation. Also importantly, these funds support training of the next generation of investigators, including graduate students, postdoctoral researchers, and other research and clinical personnel. So these are the people that are going to be applying for these grants as principal investigators in 5 or 10 years' time. At the end of the granting period, a final progress report must be submitted to MDC to help track the impact of the MDC-funded research. And importantly, MDC also coordinates with investigators to ensure knowledge gained from these studies is relayed to the community to maximize impact. And this is through hosting webinars or publications or pamphlets or things like that. Also importantly, these funds provided by MDC generate new knowledge that can then be leveraged to get larger grants from other organizations such as CIHR, the Canadian Institutes of Health Research, to further build upon the groundbreaking work initiated using MDC funding. So in short, MDC supports research, training, accountability, knowledge translation, and future investment in research. So it's a very crucial function in the Canadian research ecosystem for supporting research. And with that, I'll pass it back to Amira.
Thank you so much, Dr. Parks. We really appreciate your ongoing support with the research grant competition, with the Medical Scientific Advisory Committee. And I also want to take the opportunity to go ahead and thank, I should go to this slide here, to thank we had 17 peer reviewers involved. We had 6 community or lived experience readers involved in our grant selection process. And really, these are individuals who are giving their time, volunteering for the organization, really sharing their expertise so that we are selecting the best science. Thanks again, Dr. Parks. With that, I'm really excited to go ahead and recognize and announce the recipients of our Translational Science Seed Grant recipients, and you'll hear from each of them today. I'd also like to go ahead and share the recipients of our Clinical Research Grant recipients, and again, you'll hear from them today. And without further ado, I'll go ahead and pass it over to Dr. Corinne Chauquet, who will be sharing a flash talk on the awarded study. Over to you.
All right, can you see my slides properly?
Looks great.
Okay, awesome. So first I want to say a big thank you to Muscular Dystrophy Canada for awarding me this grant. And this is especially meaningful because it's my first grant as an independent investigator. So my team and I are really excited to study pre-mRNA splicing to improve therapeutic avenues for dysferlinopathies. And before I get into dysferlinopathies, I just want to tell you a little bit more about pre-mRNA splicing. So as you might know, DNA is where our genetic information is stored. And you can think of DNA as a large book that contains thousands of paragraphs. And each of these paragraphs or genes encodes or contains the information necessary to make one specific protein. And proteins are the building blocks of our cells. But there's actually a few more steps between DNA and protein. So first, each of the paragraphs in DNA must be individually copied into something that we call pre-mRNA. And looking at this, you might say it's just a jumble of characters that don't make any sense. But if we start to look more closely, we might recognize some words that actually do make sense to us. And so splicing is the process by which the parts of the pre-mRNA that don't make any sense are removed, and the words that do make sense are pasted back together, such that we get a mature mRNA that contains only intelligible words and that forms a sentence that makes sense and that can be read out to form protein. Now, in genetic diseases such as muscular dystrophies, there's generally an error in the DNA. And this error gets copied into the pre-mRNA and the mRNA. such that now our sentence does not make any sense. And this is usually accompanied by the absence of protein or the formation of dysfunctional protein. And this is typically what leads to the symptoms that we can see in muscular dystrophies. So over the last few decades, there's been tremendous advancement in something called splice switching therapies, which will use antisense molecules to completely skip the word that contains a mutation or an error. And so when splicing happens, this gives us a sentence that is slightly different, but that makes sense and that means something. And this can lead to the formation of a partially functional protein that can be associated with milder symptoms. So in addition to SMA, which we talked briefly about, this type of splice switching therapies have been approved, or there's 3 of them that have already been approved by the FDA for Duchenne muscular dystrophy. And so we're interested in seeing if this can work for other types of muscular dystrophies, and especially for dysferlinopathies, which are a group of muscular dystrophy and myopathies that are caused by mutations in the gene DYSF. So there's already several groups that have attempted splice switching therapies for dysferlinopathies. And they've had some success at skipping the word that contains a mutation. But this was often not accompanied by the production of partially functional protein. So our project aims to understand the discrepancy between these 2 readouts. Now, oftentimes when researchers look at splicing, they focus on the area of interest. So in this case, where the mutated word should be. And this tells us whether we were successful in skipping the word or if it's still present. But in fact, through my research, we've found that splicing of one word can significantly impact splicing of other words in the same mRNA. So by looking only at the area of interest, we might miss some splicing defects, such as the retention of some characters that make it into a sentence that doesn't make any sense. And this could lead to the absence of functional protein. This is especially relevant in the context of DYSF, which is a really long sentence that contains 56 words or exons. And so we use a really cool new technology called nanopore sequencing that can read DYSF from start to finish instead of looking only at specific areas. And so this will enable us through this project to really study how splicing each word can influence splicing of other words. So for now, we're doing this in healthy muscle cells, and we're testing different antisense molecules against 8 exons or words that are known to be mutated in dysferlinopathies. And so for each of these treatments, we'll look at how it impacts the localization and abundance of the mRNAs, and at whether antisense molecules affect more than only the target exon. And if that's the case, how can we fix it? So in the longer term, we're hoping that the knowledge gained from this project can be translated into patient cells and to other neuromuscular disorders that would be amenable to this type of therapy. And we're looking forward to contributing to improving therapeutic options for individuals living with neuromuscular disorders. So lastly, I want to say thank you again to Muscular Dystrophy Canada and to you for listening today.
Thank you so much, and congratulations on your project. We're really looking forward to the outcomes of the study. Next, we welcome Dr. Menzies from the University of Ottawa for his study, a new in vitro eccentric muscle contraction assay for drug repurposing for muscular dystrophy.
Right, can you see that? Fantastic. Yeah, I'd like to start off by thanking Muscular Dystrophy Canada for providing the funding that otherwise this project would not be able to go forward. So thank you for that. The project is called Using Muscle Cells to Screen and Repurpose Existing Drugs to Treat Neuromuscular Diseases. I'm Keir Menzies. I'm the lead PI, and my postdoc, Alex Green in my lab, is the co-principal investigator. And then we have some collaborators from the University of Ottawa, Mike Delicio, Junie Medort, and also a collaborator from France who's providing patient muscle cells. So the problem that we're interested in is the loss of functional dystrophin in Duchenne's muscular dystrophy, but as you'll see, this project can be applied to other neuromuscular diseases. And so in this disease, there is a susceptibility to muscle fiber damage due to the lack of dystrophin. Without going into all these specifics, what it ultimately means is that with contractions of our muscles, we eventually get the breakdown of muscle in DMD. And that eventually leads to increased inflammation and fibrosis and the chronic loss of force in muscle. In particular, lengthening contractions are the worst type of contraction. Great if you're very healthy, but not so great for muscular dystrophy. And that's the type of contraction where if you're running downhill, the top of your muscle, the quads, as they lengthen, as they try to slow you down and contract, that can create a lot of muscle damage. So we're going to use that in our project, and I'll show you later how. So this current standard of care is corticosteroids. It helps to maintain strength and function in patients, delays cardiomyopathy, but there's an adverse long-term effect, which ironically is muscle wasting, fractures and weight gain. So therefore, we hope to identify new drugs that don't have these adverse effects. And how can we do this quickly? So the idea is that we will use drug repurposing, which is essentially taking existing modifications, medications that have established safety profiles that could potentially help DMD patients, and then see which of those might work out in the project. So essentially what we're using is a drug library. It's a unique collection of thousands of FDA-approved drugs for high throughput screening. And they've all been well characterized for their activity and targets and safety profiles in humans. But how are we actually going to use that library of drugs? So we're going to perform a unique screen, which I'll describe down below, that will help identify drugs that can either prevent muscle injury or boost repair in human DMD patient cells. And then we can work on to looking at other types of patient cells using the same mechanism. So the design is combining a 3D patient muscle. So we get these muscle cells from patients, and then we can form them into a muscle tissue that can contract. And so this is representative of a cell culture well. So these are plates where we keep cell culture. Sorry about that. And so inside that well is media that keeps that muscle healthy and gives it all the nutrients it needs. And then what we can do is we can contract it using electrodes. We use electricity to contract it, but we can also, at the exact same time, lengthen it, much like that lengthening contraction that you would experience when you're downhill running. So this is the apparatus that would lengthen the muscle tissue. So if we zoom into a well, so that's a circle, this is looking at it from the side. This is the muscle tissue. And then using a vacuum, it can stretch a membrane that the cells are on. And then those cells, you can see they're flatter here than they are here. And so they stretch out, and that creates the lengthening. And at the same time, we can contract. And so this can look a little bit like this. And so you can see there's a slight contraction. This is just 20% contraction. We can all adjust that to be different amounts of lengthening and contracting at the same time. Ultimately, what we're going to do is we're going to take many of these wells and test each of these wells with different drugs to define what types of drugs can help minimize damage or help with the repair of tissue using cell culture from DMD patients. We can also just look at how this mechanism of lengthening contractions alters cells in general, and how do they respond to damage, again, gene expression and protein expression in that circumstance. So ultimately, the outcome is to identify approved medications that can prevent or repair muscle damage to treat DMD symptoms. And this may prevent the need for corticosteroids or may complement corticosteroid treatment. And the advantage for DMD patients is that we can identify great drugs that can be used as candidates for rapid clinical repurposing. So use them in patients sooner rather than later. And then the other advantage is we can use the same apparatus for many other neuromuscular diseases. So it can be quickly modified just by using different types of cells from different patients and doing the same thing. and testing different drugs to see their efficacy. So I'd like to thank Muscular Dystrophy Canada again. And if anybody would like to contact me, here's my email address, kmenzies, or Alex Green, Ottawa. And there's a picture of our lab. And so thank you very much.
Thank you very much and congratulations. Next, we'd like to welcome Dr. Lisa Hoffman from Lawson Research Institute, who'll be speaking about her project, Microdystrophin Replacement Therapy for Duchenne Muscular Dystrophy.
Thank you so much for the introduction, Hamira. Can you hear me? Yes, okay. I'd like to start by thanking Muscular Dystrophy Canada for awarding me these funds and for inviting me to speak here this afternoon. I'd also like to acknowledge and thank the people on my team, co-applicants and collaborators who make all this work possible, including Drs. Craig Campbell, Jeffrey Chamberlain, and Derek Rancourt, as well as members of my laboratory, Dr. Margarita Egian, Andrew McLennan, and Rima Flynn-Risk. Finally, I'd like to thank Stem Cell Technologies for their continued support of this project. Okay, so Duchenne muscular dystrophy is a severe X-linked neuromuscular disorder caused by loss of function of the cytoskeletal protein dystrophin. It is characterized by progressive skeletal muscle degeneration. Sadly, there is no cure still. Thus, the need for effective treatments for DMD is abundantly clear. At present, the DMD field predominantly utilizes approaches aimed at replacing dystrophin via gene therapy, as in this proposal, cell therapy, exon skipping, CRISPR technology, etc. However, by the time boys afflicted with DMD are diagnosed at about 3 to 5 years of age, ischemia, chronic inflammation, and as shown here, fibrosis are profound, generating a microenvironment that is a major impediment to muscle repair. So at present, few studies are aimed at enhancing this muscle microenvironment as a means to aid muscle repair and to enhance the efficacy of therapeutics such as microdystrophin replacement. Excitingly, we've now demonstrated that levels of vessel maturation stabilization factor angiopoietin-1, or Ang1, are significantly lower in DMD mice than in healthy wild-type mice. Importantly, localized administration of Ang1 significantly enhances muscle perfusion, reduces inflammation and deposition of fibrotic tissue in DMD mice. Delving further into the effects of Ang1 treatment, we've now shown that both myofiber perimeter and microvessel circularity are significantly increased in Ang1-treated DMD mice. So given these findings, we hypothesized that Ang1 delivery to skeletal muscle will diminish inflammation and reestablish a microenvironment in which muscle repair can exceed fibrosis development and aid in microdystrophin replacement. The specific aim of this project is to assess the ability of Ang1 to enhance microdystrophin replacement therapy in human DMD skeletal muscle organoids. To achieve this, we will use a well-described three-dimensional human-induced pluripotent stem cell-derived skeletal muscle organoid culture protocol, in which muscle organoids exhibit characteristics of differentiated myofibers shown here, including organized contractional machinery. The organoids are further comprised of undifferentiated cells, endothelial cells, and mural cells such as pericytes, and smooth muscle cells that are capable of forming vessels. To address the efficacy of our treatment, we will use an in vitro assay based on the assessment of sarcolemmal or membrane damage after repetitively applied electrical stimuli. We expect that human DMD skeletal muscle organoids will exhibit a more significant increase in membrane damage following electrical pulse stimulation than healthy control organoids. We also expect that microdystrophin replacement will reduce membrane damage. Co-application of Ang1 will further enhance the efficacy of microdystrophin, improve vascular integrity, and attenuate ischemia. So, over time, treatment challenges in DMD multiply due to marked ischemia and fibrosis in skeletal muscle. With our Ang1 approach to slow or attenuate these conditions, we believe and hope that the benefits of therapeutics from DMD may indeed be fully achieved. Thank you.
Thank you, Dr. Hoffman. We're really excited about the results of this study, and I remember our lived experience readers really looking forward to the outcomes of the study, really timely when we think about Duchenne therapies. Up next, we'd like to welcome Dr. Rashmi Kothari from the Ottawa Hospital Research Institute, who was awarded funds for his study, Maternal Transfer of AAV Vectors, a minimally invasive approach to deliver SMN gene therapy for SMA.
Thank you, Amira, and thank you, Muscular Dystrophy Canada, for the continued support of our work. Unlike Karina, my support goes back many years and decades, in fact, from Muscular Dystrophy Canada. So much appreciated for that. This particular proposal that was just recently now funded is maternal transfer of gene therapy for spinal muscular atrophy. And although I am the PI, principal investigator on this grant, I wanted to acknowledge the strong contribution of the co-PI, co-principal investigator, Dr. Emma Sutton. Emma is a postdoctoral fellow, recently arrived from England. She has been in my laboratory for a year and has developed this project entirely on her own efforts, and I really think she's going to do really well with this study. Let's see. So spinal muscular atrophy, I think Robin Parks introduced this a little bit. Its short form is SMA. It's a genetic motor neuron disease that's characterized by motor neuron death, and that leads to muscle atrophy. And that's just in a very basic and simple manner is what spinal muscular atrophy is. It's a disease of the motor neurons, although other organs and tissues are implicated in the disorder. This degeneration of motor neurons and muscle atrophy leads to progressive muscle weakness, as indicated in the pictures. And then children very early in life have difficulty breathing and feeding. And so the severe form of SMA is, unfortunately, is fatal within the first couple of years of life if left untreated. In Canada, there are 700 to 2,000 people at any one time living with SMA, and newborn screening estimates now about 35 new cases per year. So it's a rare disease, but nevertheless a very important one to treat. And in many ways, SMA is a model disease for neuromuscular diseases because we have had tremendous progress, as again articulated by Dr. Parks, over the many decades and years of clinical research, followed by basic research, and then preclinical to translational research. There are development of not one, not two, but three treatments for spinal muscular atrophy. These are now FDA approved and available in the clinic. The first one is an antisense oligonucleotide from Biogen. It functions to make the copy gene, the second gene, SMN2, behave like the more important SMN1 gene. The second approach is a gene therapy approach, where adeno-associated viruses, these are related to the flu virus, are used to deliver the wild type or proper gene to replace the loss of the SMN from patients. And the third approach is a more recently approved therapy. It's an oral medication, but that's called a small molecule, so it's really in a pill format. But it does a similar function as the antisense oligonucleotide approach in that it tricks the SMN2 gene to think that it's the SMN1, and that therefore produces the good protein for therapy. So really, we have lots to be thankful for in the sense that we do have these therapies, but there are limitations. Not all patients experience a large benefit. And for many of these therapies, it's probably because we're treating the patients after birth, meaning that by the time we get to know which patients need the therapies, they already initiated some of the pathogenesis, and therefore there is no going back, and therefore the response may not be as effective as one would have hoped for. And so what we are also discovering is that SMA may have developmental issues that begin in gestation during pregnancy. And if that's the case, then really treating babies after birth will have a lot of limitations. And finally, really, currently, none of these therapies is curative. So the unmet needs as far as the therapeutic field has to face are that currently treatment for SMA is given to babies after they're born, and that's the best we have now. Fortunately, have newborn screening in place, which allows for identification of babies very early after birth. But nevertheless, the earliest you can get to these babies is 2 weeks to 4 weeks after birth, and that may still not be early enough. So we aim to find a new way to treat SMA in babies before they're born. And if you can do this, that would be quite striking. So really what we're suggesting is we would like to see if it can work better if we give the therapy to the baby's mother while she is pregnant. So deliver the therapy, in this case we're studying gene therapy, to the mother rather than wait for the baby to be born. And as always, we'll begin our studies using mice, our preclinical model. We have developed mice that have all the features of spinal muscular atrophy, and we'd like to see if giving the treatment to the mother can stop the disease from causing problems in the baby. So using mouse as a model system will really allow us to tackle this question very thoroughly. And so here's what we're going to do. Shown on this slide on the first panel is the what we call AIM-1, or the first experiment that we hope to do, and it's called maternal transfer of AAV. This is the least invasive approach, where we simply inject either the muscle or into the tail vein or vein, to the blood system, the virus that will then be delivered throughout the pregnant mother, and hopefully we hope that it traverses the placenta and gets into the fetuses, which will then incorporate the good gene into their cells. And then after birth, these babies will then be examined to see how effective this maternal transfer was. And that's the first part of our study. The second part of our study, which is AIM-2, is to do a little bit more invasive approach, which is called in utero or intraplacental injection. So this requires getting the injection and getting access to the developing fetus, where we would inject the placenta with the virus and thereby hope that it gets delivered to the fetus and all the cells of the fetus. And we will examine the mice after they're born to look at the success of this approach. And in the really long term, we are quite hopeful and that this research could mean that treating the mother before the baby is born might be a new and better way to treat SMA patients. And we're using gene therapy as a model, as a way to do this, but we could think about this in a similar fashion using the risdiplam, which is the oral medication, so delivering that, or the antisense oligonucleotide going forward. Finally, just thank the people that do the work. Emma Sutton leading this study in the lab with help from a lot of other people. Our collaborators, Bernard Schneider in Switzerland, who works with us to produce and develop AAV, the gene therapy vector, and of course, MDC, Muscular Dystrophy Canada, for their continuing support. Thank you very much.
Thank you, Dr. Kothari. That was a really clear and well-delivered talk, and I think it really highlights a point that was made by Dr. Parks earlier in terms of investing in that translational science. Sometimes it's not an easy way to understand how that basic research really translates to humans or the clients that we hope to serve and support. But this is a clear example of that research. It also reminds me of a recent work that actually came out of Ottawa Hospital as well, which was the first in utero, first in the world for another neuromuscular condition in terms of Pompe disease. So really hopeful in terms of seeing the results of this work. Congratulations.
Thank you.
With that, I'd like to go ahead and welcome Dr. Jerry Pfeffer from the University of Calgary to present his proposed study.
Thank you very much for the chance to present our work, and nice to see all of you. My project from our team is looking at single nucleus RNA sequencing in adult onset muscle diseases. We're doing this project together with Dr. Sam Chipper, who's one of my neuromuscular colleagues in Calgary and a longtime collaborator, and members of my team, Dr. Tyler Soule and Matt Joel. And I first wanted to start by saying thank you to Muscular Dystrophy for supporting this work. And I also wanted to mention I've really enjoyed the presentations that have come for this. I feel very honored to be presenting among this group of accomplished scientists. So thank you. So our project focuses on patients with adult onset myopathy. And part of the reason that we've chosen to focus our research on these patients is firstly that this is what I specialize in. Most of the patients in my clinic have adult onset myopathy. And this is when people develop muscle weakness at some point in adulthood after a long period of normal muscle function. And these conditions are very difficult to diagnose. We don't have very many specific biomarkers for these conditions, and they are in many cases of unclear etiology. And I'll point you to this figure from a paper that surveyed genetic testing for myopathy across Canada. And in the adult cases, effectively, they have a diagnostic hit rate of around 30%, and almost 70% have no diagnosis. So that is the group that we are focusing on in this project. I think it's really important to get some kind of diagnosis for everybody. We've seen in some of the previous studies that in conditions where we have a molecular understanding of the disease, that we're eventually able to make therapies and improve their condition. But as long as we're stuck with a large group of undiagnosed patients like this, it's going to be a lot harder for us to find things that work. So we wanted to focus on this group. Is there a way to improve genetic diagnosis? Or if some of these cases are actually non-genetic, relating to environmental factors or other complex genetic factors, is there some biomarker that we can identify in order to help select out these cases? So this project builds on previous work that we've done in our lab. We've been interested in RNA sequencing in muscle disease for some time. And I'll point you to this work that was done by Matt Joel from my lab. And in this figure, we're showing you a principal components analysis with patients who are diagnosis resistant, so did not have a genetic diagnosis, but have the following characterizations on their muscle pathology. And you can see that in general, diagnoses of the same pathology tends to cluster together with regard to their transcriptome signals. And specifically, when you look at mitochondrial myopathy, for example, you have very particular signals that stand out that are very well characterized when looking at muscle RNA. One of the main criticisms or areas for improvements, though, that we got from this study was, well, you're looking at the muscle sample as a whole. And a muscle sample includes muscle cells, but it can also include immune cells or connective tissue cells or stem cells. And they might be in different quantities from one sample or another. So how do you address that? How can we do better? So we worked on a method to look at single nucleus RNA sequencing in muscle tissues. And specifically, we worked to optimize this method for human muscle tissues that were in storage from biopsy procedures. And this is very nice work done by Tyler Soule from my lab, which was published last year. And in this study, we were able to show that you can separate out different types of muscle cells based on their differing transcriptome signals. Here, for example, type 1 muscle fibers, type 2 muscle fibers, and a cluster of differentiating muscle fibers in between, and various other cell types of interest. And on the right, there's a heat map. Of course, any discussion of RNA sequencing needs to include a heat map by definition. And you can see that based on the transcriptional signature, we're able to identify the various different cell types. So for this project, we've applied to use this approach, looking at patients who have diagnosed muscle disease with specific genetic subtypes and cases that are diagnosis resistant, meaning that they've had extensive genetic sequencing, but we have not been able to reveal a diagnosis. And we're going to try and identify signals either by differences in the cell types, differences in inflammatory or differentiating cells, in order to divide these up into groups that can be studied further to better understand their underlying causes. So we are hoping to identify some adjunctive test in muscle that will help us find signals that are specific to the genetic versus the diagnosis resistant cases. We're hopeful that this will provide some information regarding pathogenesis in these undiagnosed cases, which again make up 70% of adult onset myopathy patients. And we're hoping that this could potentially be translated to a biomarker panel in the future. And I just wanted to give thanks one more to my team. So this is Tyler Soule over here and Matt Joel over here, and the rest of my team, who are all awesome and who make sure the work gets done. Thank you very much again to Muscular Dystrophy Canada for supporting this work. Thank you.
Thanks so much, Dr. Pfeffer. This is really important work, and I want to highlight one key point, which is we have a number of clients who have exactly that, adult onset myopathies without a known variant or a known neuromuscular condition yet. And so work like this will really help to ensure people have a firm diagnosis and they're really not stuck in that diagnostic odyssey for a number of years. So really important work, and congratulations. With that, we'll go ahead and switch now over to our clinical research grant recipients. And first, I'd like to welcome Dr. Abenheim from Sir Mortimer Davis Jewish General Hospital, and he'll be speaking about maternal and neonatal outcomes of cesarean deliveries in women with muscular dystrophy. Over to you.
Thank you so much, Homira. Do you guys hear me okay? Okay, great. So I will share my screen. First of all, thank you very much for giving us the opportunity to present our proposal and for funding this work, which we feel is quite important and will allow an increase in knowledge that can have a direct impact to those women with muscular dystrophy who are pregnant. I'll give you a little bit of background of our team. Switch this here. Okay, so our team, we have OBGYNs, maternal fetal medicine specialists, epidemiologists, biostatisticians, and carry out the research component of the work that we do and provide the methodology expertise, the clinical expertise, and carry out our projects. Our focus of our research team is prematurity, rare diseases in pregnancy, in which this is the one category it falls under, cancer in pregnancy, and epidemiology and clinical research. So much of what we do is directly related to patient care and centered over patient care. Our funding comes from CIHR, Canadian Cancer Society, and specific organizations such as this organization, which is funding this important project. I'll give a little bit of background as to the work that we have previously done, and that is a paper that we published on obstetrical, maternal, and neonatal outcomes in pregnancies affected by muscular dystrophy. One of the important findings that we saw is that there's an increasing number of women per 100,000 in the US that have muscular dystrophy and are pregnant. This used to be a much smaller number, but we're seeing more and more women are pregnant with muscular dystrophy, and there's a big gap in knowledge as to what should be done to help these women and to know what is the best course of management for them and their pregnancies. And so our research focus is always on what happens with these women that are pregnant and what we can identify predictors of outcomes. When we looked at their outcomes in pregnancy, we saw that there's a much higher rate of many adverse pregnancy conditions, such as preeclampsia, preterm labor, early rupture of membranes, infections, and postpartum hemorrhage weren't much, but we saw a considerably higher number of cesarean deliveries in women with muscular dystrophy, as one would expect, but also more instrumental deliveries. We also saw considerably higher risks of serious adverse outcomes, such as blood clots, venous thromboembolisms, a need for transfusion, and cardiac dysrhythmias, which are conditions that are quite serious, and we have to be prepared and ensure that women with muscular dystrophy are in the proper centers where they can get adequate care and also have their conditions recognized early so that adverse outcomes don't progress to more serious conditions. We also found that there are more problems in the newborns, which tell us that we need to be prepared to ensure that the proper newborn and neonatal teams are present at the time of the delivery, and for the postnatal care. More prematurity and more growth restrictions, all are things that needs to be properly monitored with adequate surveillance and serial ultrasounds. Our objective of our study was really based on the consideration that in our previous work, we found that 40 to 50% of mothers with muscular dystrophy have cesarean deliveries. And we wanted to evaluate the effect of these deliveries on the pregnancy and on the newborn, and particularly on the outcomes surrounding the delivery for the mother. We wanted to see what are predictors of adverse outcomes. Can we identify features, clinical conditions in the mother that can predict these adverse outcomes so that we can be ready and we can prevent these things from happening? And we also, more importantly, wanted to be able to identify those women who should be delivered in tertiary care centers. There's a lot of push to have deliveries more in a natural environment, and some people are pushing for that and being advocates for that. And I think that's great if we can identify those women where we can ensure that their deliveries can occur safely, and which ones should really be delivered in tertiary care centers with the adequate resources. The design we're going to use is a retrospective design using a large cohort. We've done a lot of work with this database called the Nationwide Inpatient Survey, where we were able to get over 8 million births in the United States with a large list of diagnoses and procedural codes that had occurred during their delivery admission. And with that, we can identify those births occurring to women with muscular dystrophy, and at the same time, we can also try to see what kind of interventions were undertaken and what are predictors of those adverse outcomes, so that we can aim to identify them, know what they are, and prevent the adverse outcomes from occurring. And once we identify those pregnancies, we can carry out our study objectives. Our long-term goals is to increase knowledge of the condition in pregnancy. And this is an understudied area. Like all rare diseases that we study, there's an enormous gap of knowledge in the literature. And so because not all births occur in the same hospital where people can develop expertise, this is pretty much scattered all over the United States, all over Canada, and you want to be able to, when a person, a high-risk OBGYN gets a case of muscular dystrophy, they will invariably go look at the literature and see what they should know about it. And that's the knowledge that we're hoping to provide those care providers. Hopefully, the knowledge that we will acquire will provide information that will enable safe outcomes for the mother and the newborn, irrespective of where they are. And that's really the key thing. It'd be great if all people with a certain condition were in one hospital where that expertise could be available, but in reality, that's not the case. We want to be able to provide that knowledge all across for wherever that patient is. They will be empowered with that knowledge, and they can also provide the providers with that knowledge, but in the era of up-to-date and availability and access to resources, the more knowledge we push into that data, into that in the resource pool, the more these women will be able to have adequate and proper care for their pregnancies and their deliveries. So thank you for that opportunity.
Thank you so much. That was a great study, and we're really looking forward to these results as well. And actually a really good example of, while we want to support projects that will help to inform treatments and therapies and diagnostics, but in this case, really supporting that clinical care and the management of patients affected by neuromuscular conditions. Wonderful. With that, I'll go ahead and call up next Dr. Krista Best from Laval University, who will be speaking about her project, Efficacy of Wheelchair Skills Training to Improve Mobility for People with RSACs and Myotonic Dystrophy Type 1.
Okay, good. Thanks. So thank you, Omira. Thank you, Muscular Dystrophy Canada, for supporting our project called Efficacy of Wheelchair Skills Training to Improve Mobility with RSACs and Dystrophy, dystonia muscular, myotonic dystrophy. Now I'm thinking in French and speaking in English. MD1. I'm going to tell you what it is in a second. Our study team is comprised of a group who really know a lot about RSACs and MD1 that I'm going to describe in just a second. Dr. Cynthia Gagnon is a researcher and physiotherapist at University of Sherbrooke. Dr. Xavier Rodrigue is a physiatrist at the center where I work in the CIRIS. And we also have Isabelle Lessard, who's a physiotherapist at CEGEP Jonquière. They're really experts in neuromuscular disease, including we also have expertise in wheelchair mobility. So Dr. Francois Routhier from University of Laval and CIRIS, Dr. Lee Kirby from Dalhousie University, and Omir Osman, who is our knowledge translation expert, who's going to help to make sure that we mobilize this knowledge in the appropriate way for the population. So our project is really focused on treating or looking at ways to improve mobility for people with neuromuscular disorders. And we decided to focus on two disorders specifically, RSACs, autosomal recessive spastic ataxia Charlevoix-Saguenay, and myotonic, here we go, myotonic dystrophy type 1, MD1, which are rare neuromuscular disorders, but they're quite prevalent in the province of Quebec. So RSACs is a rare hereditary degenerative disease with neurological impairments that progressively reduce mobility, and MD1 is a multi-system disorder that affects skeletal muscles and smooth muscles and also the central nervous system. I also want to note that in this project, we will be open to other neuromuscular disorders that are recognized by Muscular Dystrophy Canada, because wheelchair mobility really applies, and the approaches we use to do wheelchair evaluation and training can apply on a large scale. I'll talk a little bit about that. So what we know about wheelchair mobility is that it has positive impacts on mobility, helping people get around, also on social participation, and it can reduce caregiver burden. The challenges of wheelchair mobility are that there's often acute and chronic injuries associated with wheelchair mobility. There's often dependence on others for mobility, which can lead to reduced social participation. And in a longitudinal study over 5 years with a population with RSACs conducted by Cynthia Gagnon, we found that about 45% of adults with RSACs use a wheelchair for mobility. So given its progressive nature and decline in mobility, people often start using wheelchairs around the age of 25 to 30. What we also found in this study is that there wasn't a whole lot of follow-up regarding mobility after a wheelchair was prescribed. So I'll give you a little bit of a portrait. We've been conducting wheelchair mobility research for myself a little over 20 years, and a program that I'm going to introduce in a minute, since 1996. So this is a portrait that we've put together based on various types of wheelchair users, various ages. And what we know is that wheelchair skills capacity, that's somebody's ability to use their wheelchair, and varies from about 65 to 85%. The performance, so that's how people actually use their wheelchairs, the things they do, is about 55 to 70%. And confidence for using wheelchairs is between 65 and 90%, depending on populations. In this longitudinal study that I mentioned, we looked at what is the wheelchair mobility of people with RSACs. And what we saw is that it's quite a bit lower than, I'll say, the general population of wheelchair users that we've been collecting over the last 20 years. And we saw that wheelchair skills capacity was about 46%. Performance was also about 46%. And the confidence was between 58 and 73%. And that was in a study with about 40 people with RSACs in the province of Quebec. When we talk about prescribing a wheelchair, providing a wheelchair, the World Health Organization talks about 4 steps that we should follow. And these steps are select, which includes evaluation and finding the right wheelchair, then also finding the right fit to the person. Also, it should include training, so teaching people how to use their wheelchair, and then a follow-up. So after the person gets a wheelchair, gets some training, there should be some follow-up, 3 months, 6 months, 1 year, maybe even years after. So this project is really going to address 3 of the 4 steps: to select, train, and follow up. I mentioned this wheelchair skills program that we've been working on since 1996. It started. Personally, I've been working with the program since 2002. And we've conducted lots of different research looking at how to better evaluate and train people to use their wheelchairs to help them get around, to improve mobility, to improve performance, which can also enhance social participation or taking part in the activities that are important. The website is there also for anyone interested. What we know from various studies that we've done over the last 20 years is that wheelchair skills training following the program increases wheelchair skills and confidence. The biggest thing to know about these 2 graphs is anything on the right-hand side of that orange line means that it works. So in most of the studies that we've conducted, we see that wheelchair skills training works and improves skills and confidence. We did a little bit of pilot work from that longitudinal study that I mentioned with 3 participants who had RSACs. They were 3 males between the ages of 40 and 49 years, and they had 10 to 13 years of wheelchair experience already. And they did 5 45-minute wheelchair skills training sessions in the community. What you see here is some of the obstacles that we trained to overcome. So you see some gravel and grass to get across. You see a bike path to get through that had a bit of an incline, a little bit of a curb level change, a long hallway with carpet, some irregular surface, long ramps and gravel. And from that study, what we saw, if you look at the green columns, there was an increase in wheelchair skills capacity, so their ability to do that in all 3 participants that varied between 10% to 36%. Confidence also increased to 5% for 2 participants and 15% for another participant, but their perceived performance went down. We have some ideas of why that I won't get into yet. I'll save that for sharing the results of our study. So the aims of our study are to evaluate the wheelchair skills program, so to conduct wheelchair skills training for people with RSACs and MD1 to see how we can improve manual wheelchair skills and confidence. We're also going to conduct some qualitative interviews to explore what are expectations and experiences with wheelchair skills training. And then we're going to follow up with people 3 months later, because we know that changing behavior and we know that learning skills take some time to apply. So we want to see how people are doing 3 months later. What we expect, what we know from our previous studies and doing some tests with our evaluation measures is that 1 wheelchair skill equals about 3% on our test. And we expect that we'll see about a 20% increase based on our pilot work and previous studies, which equals about 6 wheelchair skills. So if you think about your wheelchair skills being getting over carpet, getting up small inclines to access that bike path, or getting over small level changes to get through that apartment door, it really could have an impact of helping people to get out of their house, which could impact the ability to go grocery shopping, to see friends, or to have a job. And so we think that we'll be able to have an impact of about 5 or 6 wheelchair skills. And with that, I'll thank again Muscular Dystrophy Canada and look forward to sharing our results when we have them.
Thank you so much, Dr. Best. And we're not only happy to be funding this project, but also supporting the knowledge translation and mobilization of the study. Wonderful. Up next, we would like to acknowledge Dr. Natalie Baer, and on behalf of Dr. Natalie Baer, Samar Muslimani, who will be leading this study, will be talking about this project, understanding the impact of the central nervous system impairments on daily life in myotonic dystrophy. Over to you, Samar.
Okay, hi everyone. Are you hearing me well? And do you see my screen? Perfect. Okay, so I'll start right away. So I'd like to start by thanking, of course, Muscular Dystrophy Canada for the opportunity to present today, and of course for the support of this project, which is about understanding central nervous system impairments on daily life in myotonic dystrophy type 1. So I'm here representing, of course, Professor Nathalie Bière, but also representing my whole team. So Professor Cynthia Gagnon, myself, Marc Tremblay, which is our patient partner, and Cathy Bouchard, which is our relative research partner. Oops. Okay, so it should, okay, good. So I'll start by quickly presenting some definitions. So myotonic dystrophy type 1, so DM1 is a rare neuromuscular disease that can affect not only the muscular system and nervous system, but also many others, which is why it's called a multisystemic disease. We do have a higher number of people affected with DM1 in our region, which is Saguenay-Lac-Saint-Jean. So we do many projects with this population. I'm also going to talk a lot about central nervous system impairments, which are impairments in the following functions you see on the screen. That's about it. And daily life requires the accomplishment of everyday activities that are important to the person and help them accomplish their life roles and things like that. So on the screen, you can see that DM1 can affect many everyday activities, including, for example, mobility, which is walking, climbing upstairs, meal preparation, cleaning up, chores, things like that. So what's our project all about? So as you know, the different impairments in DM1 can influence the accomplishment of daily activities. When discussing what we've noticed, and I'm talking as a clinician, I'm an occupational therapist, but also as a researcher to be, we've noticed that when we discuss about the impact of these impairments, we tend to put the focus mostly on the physical or visible aspects of the condition, which are important. But this means that we tend to overlook the influence of symptoms related to the nervous system, which I have presented some example earlier. We noticed that little is known about the impact of these impairments on daily life in individuals living with DM1. This can lead to many different consequences. I've put you just a few examples here. The fact that there might be less services offered to the affected individuals, misunderstandings from healthcare providers. For example, a lot of healthcare providers are not familiar with apathy, which is a lack of a difficulty to initiate activities. And when we're not aware of this specific impairment that might happen in DM1, we do not treat patients the same way. So this can lead to misunderstandings. There's also, we also noticed exhaustion and frustrations from relatives of people with DM1 because by not knowing the impact of these impairments, relatives might not know when or how much assistance to give, might give too much, not enough, and then this can lead, as I said, to different problems. So thus, this project aims to understand how central nervous system impairments impact daily life. So we're going to use a mixed method study design. So we'll start with a qualitative study. So we have put the point of view of patients, relatives, and healthcare providers at the center of it. So we'll do case studies with observation of daily activities and individual interviews with patients, relatives, and healthcare providers, as well as group discussion. All of this data collected will help us select the best outcome measures to do a transverse cross-sectional study with 50 people with DM1. And we expect different outcomes, mainly that the study will highlight the most important CNS impairments, which is central nervous system impairments, to consider in future therapeutic trials. And we are doing so by putting as I said, the perspective of patients at the center of our methodology. And the study will also inform healthcare providers on evidence-based practice regarding how they assess and how they intervene with people living with myotonic dystrophy type 1. We also expect that we'll be able to provide answers to patients and relatives regarding cognitive impairments and their impact on daily life, and which will contribute to improve psychological health of all affected people by DM1. So I'll end by thanking again Muscular Dystrophy for the support. We're really happy. This is the picture where most of us are present in the same time, but it's Professor Nathalie Bière is missing. But here is our team, and we're really grateful. And I think that's pretty much it. Thank you.
Thank you, Samar. I should acknowledge that both yourself and Dr. Bass mentioned the inclusion of patient research partners in on your teams, and I think that's excellent. If anyone is interested in becoming a patient or parent research partner, we have a program here at MDC. It's called Import ND, and we help to train you and help you become ready to serve on these research projects. Last but not least, I would love to welcome Dr. Regine Rajendram from Holland's Lorview Kids Rehabilitation Hospital to give us our final flash talk for this evening. Over to you.
Thanks so much, Mira. Can you guys hear me okay? Yeah, good. Okay, I'll share my screen. You guys can see that as well. Great. Awesome. Okay, so good afternoon, everyone, and thanks so much for having me. I think I share Dr. Choquet's experience in this really being my first grant as an independent investigator, so I'm extremely grateful as well for the support of Muscular Dystrophy Canada. And I'm really excited to talk about this project, which I think is really going to transform how we practice in clinic and manage patients and clients with Duchenne muscular dystrophy. So we all know that Duchenne muscular dystrophy isn't just a condition that affects muscle strength. It really is a challenge that individuals and their families live with every day, facing obstacles that probably most of us can hardly imagine. And as DMD progresses, breathing specifically, and I'll switch slides, there you go, becomes more of a laborious process than a reflex. And it's this muscle weakness that characterizes DMD that eventually results in compromises in respiratory function and making something as natural as breathing incredibly difficult. And this is really where non-invasive ventilation, or NIV, becomes a lifeline, not just a treatment. But the current approaches to identifying whether someone needs NIV is primarily reactive. Often you're waiting for symptoms to worsen before doing a sleep study, and then following kids as they progress. And so really, as we put this project together, we asked ourselves, can we do better than this? And this really led me specifically, because my specific expertise is in AI and clinical informatics, to think about how can we use artificial intelligence to address this. And I know this is a very hot topic, but AI really is not just about robots and social media, but it can be a powerful tool in healthcare. And to be honest, it's been used in healthcare for many years, but really is becoming in the forefront now. And truly, our project aims to use AI to predict the need for non-invasive ventilation in DMD patients, hopefully before their conditions worsen, ensuring timely intervention. So really moving more towards a personalized approach. So the approach is innovative, but it is relatively straightforward. I think the slides that follow will make it seem like it's not, but essentially the process or the methodology for an AI project involves collecting data, large amount of data. And I have the fortune of working with some collaborators who are listed on the first slide, Dr. Laura McAdam and Dr. Rashma Amin at SickKids, who together, I think, sees one of the largest DMD populations in the country. So we have quite a big data set. And really mining that data to see if we can build predictive models. And this involves several steps, and I won't go through the fine details of those. But the goal at the end of each of an AI model project is to build one or a few models that can, with some level of probability, predict an outcome. And in this scenario, the need for non-invasive ventilation. And my hope, or our hope, really is that this method will allow us to be proactive rather than reactive, and hopefully improve patient outcomes and reduce healthcare burdens, because each of these steps do take a lot of our system, and there's a huge backlog for just to do sleep studies, for example. So there's many parts that can be improved upon along this pathway. So that's sort of the methodology in a nutshell. But in terms of its impact, I really do think, hopefully, when we're done this, we'll have an example of an AI model which will actually have a real-world impact in with our clients, and that's our end goal, to have this actually implemented as part of our clinical pathways. And truly imagine a world where we can anticipate the needs of individuals with Duchenne muscular dystrophy and able to provide intervention before they're in distress. And so really our hope with this project is not just about extending lives, but improving the quality of those lives. And so my hope with this specific project is not just to look at DMD, but eventually expand and use this as a proof of concept to look at other muscular dystrophies as well. But that will be down the road. As I mentioned earlier, I think this project really is going to be a testament to the power of collaboration. I think it truly brings together clinicians, researchers, patients, families. We do have family representatives on our team. We have, which I didn't list, but I will include in the slides if anyone wants to see afterwards. But of course, as I mentioned, Dr. Reshma Amin at Sick Kids and her team, and Dr. Laura McAdam at Home Bloorview and her team here. And along with, of course, the support of Muscular Dystrophy Canada, we're incredibly grateful for that. Together, I think we're really going to be able to build something that is going to be helpful for a lot of people. So in conclusion, I think our project really represents a leap forward in managing DMD, with AI paving the way for proactive healthcare rather than reactive healthcare. And I just want to again say thank you to the patients, families, donors, volunteers, and partners here today. I mean, you're really the heart of this project. So it's your support and your belief in innovation that's allowing us to push boundaries. So thank you for being part of this journey.
Thank you so much, Rakeem, and couldn't have said it better myself. With that, I'm going to go ahead and also share that this year, in addition to our research grant recipients for our clinical and translational grant competition, we also have an awardee from our off-cycle research grant program, and that's Dr. Hernan Gonareski from the Hospital for Sick Children for his study on the natural history of Charcot-Marie-Tooth disease. And that actually brings us to $1 million invested this past year towards research grants. And we're really happy to celebrate that. With that said, I'd love to pass it over to our CEO, Stacey Lintern, for some final remarks. Over to you, Stacey.
Thanks, Homera. So on behalf of Muscular Dystrophy Canada, congratulations to all of the research recipients. A genuine thank you to the chairs of the grant competition. We heard from Dr. Robin Parks, who led our translational grant review and selection process, and also thank you to Dr. Hugh McMillan, who led our clinical grant review and selection process. Thank you to you both for your time. I know that you have lots of competing priorities. We don't take that for granted. We are grateful your experience in ensuring that MDC is funding the best research that will advance optimal clinical health care and finding cures. A heartfelt thank you to the peer reviewers and people with lived experience reviewers who your knowledge, your expertise, and time is greatly appreciated. Thank you to our board, Canadian firefighters who raise millions of dollars for research year after year, our generous donors, partners, volunteers, clinicians, researchers, clients and their families, and networks, and of course our staff. And a special thank you to Dr. Homera Osman for her leadership and always due diligence in the research grant recruitment, review, and selection process. It takes many wonderful individuals and people that make up this amazing community so that MDC can invest in the best research possible. Because of you, MDC this year, as Homera has mentioned, has made an investment of $1 million in research grants alone, which is amazing and outstanding. Let's keep this momentum going. It is a transformational time for neuromuscular research, and our commitment to research is strong and growing. We invite you to attend our support and walk a role events across the nation that will help us to continue our commitment to invest in research. So please go to www.walk4md. We'd love to see you at one of our community events. You may know that MDC only receives less than 1% of government funding. So if you have the capacity to give a donation or volunteer or support in any other way, we would be grateful. Please reach out to Homira if you require additional information. And thank you to everyone who's involved and working together to make this happen, to break down the barriers for the neuromuscular community.
Thanks, everyone. Congratulations, and have a good rest of your day. Thanks, everyone.